Welcome to Ask a Protein Pro. Today we'll look at some quick tips for protein detection using antibodies and Western blotting. Let's get started. Are you detecting the correct protein? This is a really common question. First, look and see if the detected band appears at the correct molecular weight compared to a reliable protein ladder. It is also critical that we select a primary antibody that recognizes the sample species. Be sure that the selected primary antibody has proven data in Western blotting, and that the specificity is verified with appropriate controls. This is shown through various techniques, such as genetic modification, cell treatment, or relative expression. Invitrogen Advanced Verified Antibodies are tested for target specificity using one or more of these techniques. There are multiple strategies to improve low band intensity or a blank blot. For proteins of low molecular weight, try trypsin gels or higher percentage polyacrylamide gels. A membrane with a smaller pore size of 0.2 microns can help reduce the likelihood of transferring a low molecular weight protein through the membrane. For proteins of high molecular weight, try tris acetate gels, lower percentage polyacrylamide gels, or optimizing the transfer condition. It is important to ensure efficient protein transfer, which can be evaluated by staining total protein on the membrane post-transfer using Invitrogen No-Stain Protein Labeling Reagent or Ponso-S staining reagent. A faint signal can usually be attributed to low antigen concentration in the sample. Try increasing the sample load per well while being careful not to overload the gel. If it's a low abundance protein you're working with, optimize the sample preparation to achieve a more enriched sample. You can also experiment with different blocking buffers to find the most compatible formulation and avoid prolonged washing. Weak signal can also be caused by low concentration of antibody. Increasing the primary and/or secondary antibody concentration may be needed to obtain optimal signal with minimal noise. Be sure to adhere to the recommended antibody ranges for your detection substrate. It's also important to ensure the primary or secondary antibodies have not degraded. For this, you can run an SDS page along with a control antibody that you know works well. A degraded antibody appears as a smear in comparison to an intact antibody. When imaging a low abundance protein, increase exposure time to make the target band appear clearer. Use your imaging instrument's auto exposure features to help determine the optimal exposure time across your blot and adjust as needed. You can also try different chemiluminescent detection reagents with higher sensitivity. For example, the Thermo Scientific SuperSignal West Atto Ultimate Sensitivity substrate is great for low abundance proteins. As a general rule, make sure your buffers and other reagents are fresh and not contaminated or expired. It can also help to verify that any sodium azide from the wash buffer or antibody diluent is removed because azide can inhibit HRP or horseradish peroxidase. To start with, utilize protein databases such as UniProt or investigate literature to check if the unexpected bands could be reported isoforms of your protein of interest. Try to overcome this issue by decreasing the sample load, systematically reducing the primary and/or secondary antibody concentration, or increasing the number of wash steps or volume of washing buffer after each antibody incubation. Exhausted ECL, or enhanced chemiluminescent reaction, produces a white band on the blot due to high protein and/or antibody concentrations. We suggest reducing the protein load or the antibody concentration until a more optimal result is obtained. Exhausted ECL reaction can also be caused when too little volume of ECL substrate is used for imaging a blot. Follow the recommended volume for the ECL substrate to prevent this from occurring. So those are some easy tips and tricks for Western blotting. Visit our Western Blotting Education Center for more tips and tricks. Thanks for watching. See you next time.